Greetings. Today we're going to consider facts and concerns surrounding the Gene Technology Bill. This is an emerging crisis, and we need to keep ourselves informed. The Gene Technology Bill was introduced into the New Zealand Parliament on the 17th of December 2024. It introduces a low-risk classification for gene-altered organisms. It removes labeling of genetically modified foods, hands control to a government-appointed regulator, contains a legal mechanism to impose medical mandates, fails to specify criteria of risk assessment, and fails to take account of the lessons of the pandemic. The New Zealand government is deregulating biotechnology experimentation at a time when global concern about risks is rapidly increasing. An article in the Washington Post headlined, "The US could soon face a threat more powerful than nuclear weapons," saying, "Researchers around the globe are tinkering with viruses far deadlier than COVID-19. The nightmare of a biological holocaust is far from fanciful." The UK Telegraph on 21st of November 2024 headlined, "Lab leak, most likely source of COVID, says Professor Tim Spector, OBE." Spector, a Downing Street advisor and leading geneticist, points to an obvious cover-up of COVID lab origins orchestrated by governments to hide the risks of biotechnology. Professor Spector said that the case had shown that labs across the world should face more oversight and be treated with the same seriousness as a nuclear threat. An article in The Guardian headlined, "Unprecedented risk to life on Earth. Scientists call for halt on mirror life microbe research." Experts including Dr. Craig Venter, the US scientist who led the private effort to sequence the human genome in the 1990s, and Nobel laureates Professor Greg Winter at the University of Cambridge and Professor Jack Szostak at the University of Chicago, warned that synthetic bacteria constructed from mirror images of molecules found in nature could put humans, animals, and plants at risk of lethal infections. These fresh concerns over biotechnology experimentation are revealed in a 299-page report and a commentary in the journal Science authored by 38 eminent scientists. Richard H. Ebright is Board of Governors Professor of Chemical Biology at Rutgers University. He served for 16 years as editor of the Journal of Molecular Biology. He is a member of the Institutional Biosafety Committee, and he has been a member of the NIH Molecular Biology Study Section, the Antimicrobial Resistance Committee of the Infectious Diseases Society of America, the Working Group on Pathogen Security, and the Controlling Dangerous Pathogens Project of the Center for International Security Studies. So his words should carry a great deal of weight. In 2022, Ebright testified at the US House and US Senate hearings on biosafety, biosecurity, and biorisk management as follows, and I quote, "Researchers in Wuhan, as of 2018, had engineered SARS viruses to have a 10,000 times increase in viral growth in the lungs, a 1 million times increase in viral growth in the brain, and a 3 times increase in lethality in mice that were engineered to possess human receptors for the SARS viruses." Professor Ebright asked why the Wuhan gain-of-function research, which was funded by the US National Institute of Health, was not stopped when its initial results were known in 2018. No doubt you will join me in asking similar questions of our government. Why are you planning to deregulate biotechnology? In fact, multiple international authorities report more than 30 million excess deaths worldwide during the pandemic. Excess deaths are calculated by subtracting the prior trend from the current mortality statistics. In other words, at least 30 million people have died early with less than their expected longevity. To protect public health, biotechnology experimentation needs more regulation, not deregulation. Why is that? Well, let's go back to the beginning. Life begins with a whole cell. There's the nucleus containing the DNA, and then the cytoplasm with more than 40 million proteins and innumerable pathways of action, all protected by the membrane of the cell. DNA is at the heart of a network. Life develops sequentially from a single cell to 37 trillion cells in the human, but miraculously we retain a single conscious identity. No one understands exactly how this happens, yet biotech is editing the process. The risks to life are huge. The seed of everything that we treasure in life is contained inside our first human cell. Our immunity, strength, health, appearance, intelligence, growth, ambition, emotions, and our unique personal identity. Gene editing, by definition, crosses the cell membrane and alters the incredibly complicated and little understood genetic command and control system of the whole organism. This introduces a whole new class of more serious risks. An article in the prestigious journal Nature in 2021 was entitled, "Gene Therapy Needs a Long-Term Approach." And I quote, "Gene therapy trials are on the rise, but more needs to be done to understand the long-term risks associated with this type of treatment. There are growing concerns about the late adverse effects." Our government contends more exact gene editing tools, termed CRISPR, have reduced risks to the point where many GMOs can be classified as low risk and so released into the environment or consumed safely without any special identification or segregation. But an article in the journal Genome Research from 2022 describes, quote, "extensive genomic disruptions caused by CRISPR gene editing, involving genomic duplication and inversion of the target region, as well as integrations of exogenous DNA and clustered interchromosomal DNA fragment rearrangements. Furthermore, these genomic alterations led to functional aberrant DNA fragments and altered cell proliferation. So CRISPR is not exact as the government claims." So how does gene editing produce unexpected effects? Genes act in coordination with other genes to perform multiple functions. They do not act alone. Edit a gene, and this affects multiple traits and functions. Genes are multitasking. Say, for example, genes A, B, and C working together might affect the length of legs. A, B, and D might affect aggression. Edit gene A, intending to breed a smaller dog, and you might adversely affect its temperament. This is an absolutely huge, massive simplification. The complexity of genetic function is staggering, which means that off-target, unexpected effects of gene editing are inevitable. A 2020 meta-analysis, for example, found that over 10,000 gene variants influence human height. This points to significant levels of unquantifiable risk when a gene is edited. The Gene Technology Bill abolishes the current legislative requirement to identify genetically modified content on food labels. We will not know what we are buying or eating. Food labels and traceability through the food chain are standard measures to protect public health and have been so for more than 100 years. Without these, safety research is impossible. No one will know what caused adverse effects on health. This is astonishingly sinister and a removal of a fundamental right. The bill also removes the precautionary principle from gene regulation, which states that a product or procedure must be proved safe before release. The bill will allow the approval of many products without adequate testing or follow-up, which is an absolutely unbelievable mad assumption. Nor does the bill include ethical considerations relevant to animal welfare and human well-being. For medicine, the Gene Technology Bill includes the following provisions, and I quote, mandatory medical activity authorizations for a human medicine that is or contains gene technology that has been approved by at least 2 recognized overseas gene technology regulators. I'll say that again. Mandatory medical activity authorizations. And it also includes a clause, emergency authorizations, when there is an actual or imminent threat to the health and safety of people or to the environment, for example, a threat from a disease outbreak or an industrial spillage. The minister responsible for the Gene Technology Act, that's Judith Collins, will have the power to grant an emergency authorization. Well, to put these two things together, emergency authorization and mandatory medical activity, sound familiar? The prospect of mandated approval for and use of genetic interventions in any context is an assault on society and its successful functioning. We have just gone through 5 difficult pandemic years. The bill conflicts with the provisions of the New Zealand Bill of Rights, which guarantees the right to medical choice and freedom from exposure to medical experimentation. It also cedes New Zealand's sovereign control to unspecified overseas gene regulators. Why? The government suggests that our successful and profitable participation in the global economy requires biotechnology deregulation. And they are promising a huge boost to our economy. But according to a study by the US National Center for Biotechnology Information, only about 18% of biotech startups make it to the market. Even those that do, only a small percentage are successful enough to generate significant revenue. Biotech bankruptcies hit a 10-year peak in 2023. The biotech industry relies on glossy PR, often making unrealistic promises to generate speculative investment and qualify for government grants. The aim of the biotechnology sector is government-mandated use. It's a sort of holy grail of the industry. And this happened during the pandemic. And that's when excessive profits of pharmaceutical giants soared to record levels, even sufficient to create a negative effect on public debt, which will last for generations. The Gene Technology Bill specifically reinforces the notion of biotech mandates, which can drain the public purse and create massive economic imbalance. Farming. The New Zealand government says we have fallen behind other countries because our gene technology rules are too restrictive. However, there is a significant risk that New Zealand's agricultural economy and exports will be damaged if we adopt biotech deregulation. It will devalue our clean, green reputation and result in regulatory barriers. A 2023 study in the journal GM Crops and Food reported 26 countries, including France, Germany, Italy, Mexico, Russia, China, and India, 19 of which are in the EU, have partially or fully banned GMOs. Another 60 countries have significant restrictions on GMOs. The study cited the weak agricultural benefits of GMOs compared to their potential risks. It also noted lack of trust and confidence by the public in the regulatory processes behind GMOs. Our farm products could become subject to a consumer backlash, as has happened in the UK over the introduction of dairy products from cows fed a methane-reducing biosynthetic feed called Bovia, and a similar project is planned here. GE farming is quite different from conventional practice. It relies on patents, which favour higher costs for farmers and higher prices for the public in order to generate profits for the patent holders. This will encourage a style of corporate controlled farming distinctly different from the current diversified New Zealand practice. Containment. The lessons of the pandemic are clear. Genetic material is highly mobile and interactive. Genetically modified organisms, including altered crop traits, microbes and pathogens, can spread without limit. They cannot be contained or recalled or mitigated, and mistakes are inevitable. A 2022 study of the prevalence of accident occurrence among scientific laboratory workers found among the 220 participants, 99 or 45% have had accidents during their lab works. 60% of these have been exposed once, 32% between 2 and 4 times, and only 1% between 4 and 6 times, but a staggering 7%, more than 6 times exposed to accidents. Contamination. The techniques used to produce biotech products in a lab are not reproducible in upscaled commercial production processes. Commercial biotech manufacturing entails higher levels of variability and genetic contamination and offers fewer safeguards. A 2023 paper in the journal Chemosensors reports, quote, most biologic products are characterized by fragile macromolecular structures that are heterogeneous with a purity profile that varies with each batch, making them susceptible to microorganism contamination. Biosynthesis is more naturally variable than chemical synthesis, which makes it more difficult to characterize and manage properly. On the 23rd of December 2024, New South Wales lawyers P.J. O'Brien sent a letter co-signed by scientific experts entitled Urgent Request to Present Evidence of Synthetic DNA Contamination in Blood to the Prime Minister. It began, quote, we write to you with the utmost urgency regarding recent findings that significantly amplify the concerns surrounding the synthetic DNA contamination in the Pfizer and Moderna COVID-19 vaccines. This new evidence confirms the same synthetic DNA contamination found in Australian-sourced COVID-19 vaccine vials is in the blood of South Australian participants in a peer-reviewed study. So, 4 years after the COVID vaccine rollout, individuals affected by gene contamination are trying to bring the situation to the attention of government and initiate concern. And this illustrates just how far discovery of harm and legal redress can lag behind error in biotechnology manufacturing. Liability. Clause 187 of the bill, protection from civil and criminal liability, removes any responsibility for mistakes, accidents, and misjudgments. It protects persons involved from civil and criminal liability, however it may arise, for any act that the person does or admits to do in the performance of their functions or duties under this bill, as long as they appear to be acting in good faith. This is a carte blanche to act irresponsibly and dangerously. The Consumer Guarantees Act in New Zealand is part of the everyday fabric of our lives. We rely on it. It guarantees products must be safe, of acceptable quality, fit for their intended purpose, match the description given, match the sample or demonstration model. Clause 187 of the bill, which removes liability and responsibility, and taken with the lack of labelling requirements, irrevocably weakens these protections. So what projects might be approved? The bill does not specify how the regulator will assess any of the risks of any project. In the absence of objective protocols, there's an obvious danger that the regulator will become a facilitator. The pandemic shows just how far off so-called safe and effective assessments can be. The government cites Australian legislation as their model. Incredibly, the Australian Office of the Gene Technology Regulator has just rated the following project at the Doherty Institute, University of Melbourne, as posing, quote, negligible to moderate risk to human health and safety. And this is the project, again quoting, "The initial aim is to evaluate the safety and infectivity of recombinant seasonal human influenza viruses in healthy volunteers. These GM viruses will then be used to assess the effectiveness of therapeutic drugs or vaccine candidates to prevent and control influenza infection." So, the lab is proposing to make gene-altered versions of the flu and then test out the various genetic drugs and or vaccines on human volunteers over a 5-year period. Sound familiar? Given the low risk rating by the regulator, the project, which creates new viruses, is likely to be a shoo-in for a rubber stamp. There's a big difference between the words negligible and moderate used together in his rating. This points to a highly arbitrary and unreliable risk classification process. So our conclusion: the Gene Technology Bill is being introduced at a post-pandemic time when precaution should be greater, not less. Its provisions are highly permissive and vaguely constructed. Government claims of safety, economic necessity, and international alignment do not stand up to scrutiny. The lack of a labeling requirement speaks volumes about the disregard of public wishes. The idea that a government-appointed regulator can take away our food and medical choices is foolhardy and repugnant. What can we do about it? The public is being invited to make submissions to the Health Select Committee up to the 17th of February 2025 at the New Zealand Parliament website, and here's the link, and we'll repeat it again at the end. The bill is being fast-tracked over the holiday Christmas break. The Health Select Committee collects submissions but only allows for short in-person presentations and if desired and if possible, and little, if any, debate. So please write directly to your MP and call for an extended public debate, balanced media coverage, and detailed examination of actual evidence. There is absolutely no imperative to take irrevocable steps. Eminent experts are warning of next-level serious risks. The pandemic gave us a taste of what might be in store. This bill needs to be scrapped. Inform yourselves. Let your friends and colleagues know. This is a matter of serious concern for individuals and of national importance. We have reached a turning point in our cultural journey. Who are we, and what do we value? Stop the bill. The Gene Technology Bill will expose us all to unquantified risks that cannot be safely contained or mitigated. It is irrational that the government is ignoring the obvious safety lessons of the pandemic and proceeding with this bill even before the Royal Commission Phase 2 reports on risk and safety. So please write directly to your MP. For more information, you can visit thehadchardreport.com, globe.global, and gefree.org.nz. And please make a submission to the Health Select Committee, and here is the link. Thank you very much.